My name is Bob Bowden, and I work for the USDA-ARS in Manhattan, Kansas, and I'm the research leader of the Hard Winter Wheat Genetics Research Unit. Well, gene stewardship actually can be thought of in the broad sense, and it can also be thought of in the narrow sense. In the broad sense, it includes an awful lot of things that we do to try to understand the pathogen and understand the host and how we can best use the resistance genes. And so gene stewardship actually starts with integrated disease management. The most important thing that we can do is to keep the population of the pathogen small. We need to get rid of the very susceptible varieties, the things that we call rust suckers that are a source of inoculum. We need to break green bridges that allow the pathogen to overwinter or over summer from season to season. And we can even use fungicides to try to reduce the pathogen population. All that is part of integrated disease management. Another thing that we need to do is to know our enemy. So we can study the pathogen, in this case the UG99 rust or whatever it is, by looking at its virulence composition. We can do race surveys. We can do genomics on the pathogen and try to understand what are the avirulence genes that it possesses. And all these things are activities that members of the Borlaug Global Rust Initiative are already doing. And some people think that it's not part of stewardship, but it actually is. Another thing we need to do is to understand our cultivars. We need to understand what resistance genes they possess, and then we need to understand more detail about the resistance genes themselves. We need to understand what their specificity is, whether they have certain synergistic interactions with each other or antagonistic interactions with each other, and all these things are part of the broad sense gene stewardship concept. But then there's also a narrow sense, which is what most people think of when they think about stewardship, and that's just precisely how are we going to deploy those resistance genes into agriculture. Are we going to put them into all into one variety? That's would be called a gene stack or a gene pyramid. Are we going to distribute them in different geographic areas? Are we going to rotate them in space or time? These are all different ways that we can distribute those resistance genes and try to come up with some sort of a strategy to do it so that the pathogen is not allowed to easily evolve virulence to those resistance genes. So there are these different ways that we can deploy the genes, but in my opinion and the opinion of a lot of people, one of the best ways that we can protect the genes is by putting them together in a stack or a pyramid where in one variety we put 2 or 3 or 4 or maybe even 5 resistance genes all into the same variety. And so we call that a pyramid. And the idea there is that the resistance genes can protect each other from being defeated. So if the pathogen has a mutation that allows it to be virulent on one gene, the other genes in the pyramid can still function and prevent the pathogen from reproducing. And so that virulent mutant does not survive and spread. When we're talking about gene pyramids, the question is, what's the optimum size pyramid that we want to produce? Because the more genes that we put in the pyramid, the more difficult it is for the breeders to get all those genes together into one variety. It just makes breeding more difficult to have too many genes. So we have some discussions about what's the optimum number, and I'd say the minimum number, I would say, is 3. If you don't have 3 genes, you really do not have a pyramid. You have a platform for a pyramid, and it probably won't be durable. The pathogen with one mutation or even a double mutation could defeat something like that. The idea of pyramids is really not a new idea. We've been talking about it for 30 or 40 years. And actually a lot of the theoretical work that went behind it, the idea of using pyramids and why it should be more durable, happened back in the 70s and 80s. But at that time we really didn't have the technology that would enable us to stack the genes up. Because if you had one resistance gene in a variety, you wouldn't be able to tell if there was a second one or a third one in back of it because the first one would mask it. The pathogen would be controlled by that first gene and you wouldn't be able to tell if the second or third gene was in there. But now we have much improved marker technologies. We have diagnostic markers for a lot of resistance genes. We have new technologies, SNP chips and genotyping by sequencing that allows us to really take a close look at what's in any particular progeny for a low cost. And that's enabled us to build larger pyramids for a reasonable cost and a reasonable effort. The whole idea of gene stewardship is that we've spent a lot of effort and time and money finding these new resistance genes as a result of the Borlaug Global Rust Initiative and the great support that we've had from various agencies. And we don't want to waste the effort that's been expended. So if we do a good job of stewarding the genes, they're going to last for a long time, and they're going to be available for future generations to use. If we do a bad job, the pathogen is going to mutate to virulence and overcome those resistance genes, and then our effort will have been wasted. The whole idea of pyramids is that we want to build a way to have the resistance be durable or sustainable for a long time so that those resistance genes are available for future use. We have historically put one resistance gene at a time into our varieties, and we've done that many, many times. And what we find is that we put a resistance gene in, the variety looks great, the farmers adopt it, it becomes popular, it becomes grown on many, many hectares, and then that is a strong selection pressure on the pathogen for it to produce a mutant that can overcome that resistance gene. And so then the disease increases, the variety becomes less popular, and eventually goes out of production. And we call that the boom and the bust cycle. So there's a boom and then there's a bust as it goes out of production. And we've done this many, many, many times. And the whole idea of the Durable Rust Resistance in Wheat project is we want to stop the boom and bust cycle. We want to get ahead of the pathogen far enough that we are no longer in an arms race with the pathogen. So what we hope we can do is we can get ahead of the pathogen, we can produce a variety or at least some sort of a plan where the pathogen is no longer becoming virulent on each resistance gene and using each variety as a stepping stone to greater and greater virulence. What we want to do is stabilize the situation so the virulence of the pathogen is no longer increasing. And that's what we call durable resistance. Basically, we're trying to stop the evolution of the pathogen. We're actually anti-evolution of the pathogen. We do not want it to evolve anymore. We want to stop evolving virulence on our resistance genes. Well, the Borlaug Global Rust Initiative has been enormously successful. Their major goal was to identify new sources of resistance because when UG99 came on the scene, the problem was that it destroyed or defeated a very large number of our resistance genes. We were lacking resistance genes, and so their objective was to find more resistance genes, and it's been an enormously successful enterprise. We've found a large number of adult plant resistance genes or minor gene adult plant resistance. We've also found a large number of what we call major gene resistance. And so both of the two major types of resistance, we've been very successful finding sources of resistance. This has allowed us to be in a position to deploy that resistance in a strategic way and hopefully achieve the durable resistance that the initiative was designed to find for us. One thing that's really important is that these resistance genes are kind of a common heritage that we all share, and we all need to take care of them. We all need to be stewards of these resistance genes because if even one cultivar is released that contains an unprotected resistance gene, that provides a stepping stone for the pathogen to achieve greater virulence. So it's really got to be a community effort of all of us.